In this segment, we stay with the innate immune system, but now we move from the general overview into the actual players that do the work. So, if a patient develops redness, swelling, pus, fever, or an early response to infection, these cells and proteins are usually at the center of the story. And so, we're going to look at neutrophils, natural killer cells, and complement. So neutrophils are one of the classic fast responders, especially in bacterial infection, and they are white blood cells. They're also called polymorphonuclear leukocytes or granulocytes. And if that term sounds intimidating, we just break it down. So polymorphonuclear. Poly meaning many, morpho refers to shape, and nuclear refers to the nucleus. So under the microscope, they have a distinctive segmented appearance. Now, these cells can be produced in large numbers when they're needed, and they die off fairly quickly. There are efficient phagocytes, and so they also produce cytokines. One of the key features is that they can kind of roll along the blood vessels and uh, they sense inflammatory signals and they squeeze out into infected tissue. And this is why they're so useful in an acute bacterial defense. So they're produced quickly, they move to the problem, and they attack and signal for activation of the immune system. So conceptually, we see the bacterial signal, the cytokine cues, the neutrophil migration, and then we have phagocytosis and the cleanup process. Now, pus is partly the accumulation of neutrophils. And so when you see an abscess, that visual finding is going to tell you a lot about the immune response. If you see band neutrophils, those are immature forms that often suggests that the body is cranking out neutrophils quickly in response to an early significant infection. So neutrophils are not just a lab value, but they're actually a practical cue to what kind of process may be happening. And a physician that looks at these and tries to better understand what not only the number of white blood cells are, but what the type is, can tell you a lot about maybe the time course about how the infection is occurring. So a band neutrophil is an immature um, cell, and the body is trying to crank these out in an early response to infection. So the presence of band neutrophils indicates early infection. White blood cell count is elevated. The infection is currently developing. So immune cells proliferate during infection and then they die off when they're not needed. So when cytokine profile is not needed, these cells do not stick around. We get rid of them if they're not needed. So if you have a cut and the body response is to increase cytokines, well, neutrophils sense that the profile is maximal and they begin to seek out the infection. And pus is ultimately the accumulation of lots of neutrophils. Here we have a person with a beard, and we see an abscess. And the figure that's down in the bottom right, that's just a little bit of gauze that's been packed into the wound. And so it's just a little bit of gauze that's sticking out. But ultimately, we know that neutrophils are going to be very important in terms of a response to the immune system. So we also have natural killer cells, and these are unusual because they are lymphocytes, and they function as part of the innate immunity. So most students associate lymphocytes with the adaptive system, especially B cells and T cells. So NK or natural killer cells are a helpful exception to remember. So their job is to kill virus-infected cells and to kill certain abnormal cells, including cancer cells. So they do this by punching holes in the target cell. They deliver or inject enzyme that then triggers cell death. In other words, they eliminate the host cell that has become a problem. The body has identified infection or something foreign, and it is now trying to rid the body of that particular cell. So The concept that tends to confuse many is that natural killer cells are activated when a cell fails to present normal self-antigen, and especially class 1 MHC, which is major histocompatibility complex. So for now, let's think about the simple idea is this. A healthy body cell usually shows the immune system markers like, hey, I belong here. It's holding up a big sign that says, I'm self. I am part of you. Do not destroy me. Now, if a cell stops presenting that sign and saying, hey, I belong here, if a cell stops producing that sign, the immune system interprets that as, this is a little suspicious. You might be infected or altered or not behaving properly. And so what happens is, and we're going to return to MHC shortly, but this slide kind of sets up the logic now that helps us to make more sense of this later. If a healthy cell is presenting that I belong here message, we don't do anything with it. In the case where the class 1 MHC is reduced, this is the major histocompatibility complex, it's reduced or it's not there, the natural killer cell, it's activated. It says, something's up. So there's actually 3 types of lymphocytes that we talked about. Remember, these are the natural killer cells are a little bit different, but we also have B cells and T cells, which will be discussed in a later lecture. Now, lymphocytes kill virus-infected cells, as we talked about, by poking holes in them and ejecting the enzyme. And ultimately, the infected cell cannot maintain the normal environment. It leaks cytoplasm out and it dies. Natural killer cells are unique because, again, they are part of the innate immune response. MHC complexes, that's again something that we're going to discuss a little bit later, but again, this is where the body should be presenting self and the cell may be hiding something if it's not. It may be foreign, and this is why that matters. We need to make sure that we understand the core question. Does the cell still look like it belongs here? If not, we better take care of it. And then complement is another important part of innate immunity. But unlike neutrophils and macrophages, complement is not a cell population, but it's a group of proteins. So the job of complement is to make the immune response more effective. So they can improve phagocytosis, they can work alongside antibodies, and they can increase cytokine production, and they can help create the membrane attacks that help to prevent damaged pathogens from surviving. Now, if you want a simple description, think of complement as it's a force multiplier for cells, antibodies, and signaling. It helps other parts of the immune system do their job better. This is why it's called complement. It complements the work of immune cells and antibodies. So as helpful as it is, complement still has to be regulated. So there's a major theme in pathophysiology. Useful systems become dangerous when regulation fails. For instance, if complement activity becomes excessive or poorly controlled, it can actually contribute to disease. So even a good immune mechanism can become a part of the problem if the brakes on that system are not working. If it runs away and it takes off and now it's overactive, this is a negative thing that can cause problems long term. So certainly complement is going to enhance the immune defense, whether it's improving phagocytosis or supporting B cells. I think the key point here is that innate immunity is fast and coordinated, and it's heavily dependent on signaling. So cytokines, they direct the response. Neutrophils and macrophages attack and clean up. And then your natural killer cells eliminate suspicious cells. Complement, it strengthens the overall effect. So the system is powerful, but it depends on good regulation. So complement proteins are very helpful, and they play a positive role in immune system response. So complement, again, proteins that are going to work with other cells to help the body function more efficiently. Now, recall that diseases are caused when the function or chemicals within the body are unregulated. When complement is a positive component in the immune system, it's working well. If complement is not regulated, it may be overactive and become problematic.